As Mike suggested, I'm John Hemberger Jr., Vice President of the Geologists at Jackson Hole. Mike's done the welcome and all that good stuff, so it's my pleasure to introduce our speaker tonight, who is Dr. Ellen Currano. Ellen is an associate professor at the University of Wyoming, and she's in an interesting situation in that she is divided rather literally between the botany department and the geology and geophysics department. G&G only gets 25% of her, the botany gets 75%, and you can ask her how that was sorted out. But she is a paleobotanist. Her education includes a Bachelor of Science from the University of Chicago, and she's a Nittany Lion, a PhD from Penn State.
Penn State.
So she's a paleoecologist, and if you look at the table up here, you'll see some of what she uses, which is fossil plants, to investigate the response, basically, of climates in the past to environmental perturbations. So how did environmental changes, affect the environment? The botany, the plants and the animals and the ecosystem. So, her work, a lot of it is focused on understanding how these paleo ecosystems reacted to change in the past, which ideally will, and I know she'll talk about this here, better allow us to determine how our own modern day systems around the world will respond to man-made changes. So her research, as she said, is primarily field-based and specimen-based. So she focuses on the biotic response to climate changes during the hothouse Paleogene, one of the hottest point in Earth history, going back to the time when the dinosaurs still existed. I don't know if Mike mentioned it, but she is, our lunchtime talk tomorrow is something that she is an expert on, the evolution of East African terrestrial ecosystems over the last 30 million years. So that slide was floated past you up here, and it said something about a Lagerstätte, and basically what that is, that means it's a hell of a fantastic fossil bed. It's like the Green River down here south of us, and so she will be talking about that at lunchtime tomorrow at the Jackson Hole Senior Center. So we encourage you to think about coming to that around noon, 12:15. And then she uses fossil plants to reconstruct the past, climates and environments. So without further ado, I'd like to ask you to join me in welcoming Ellen Currano.
All right, thank you so much, John. Can everybody hear me okay? Okay, awesome. So I have been working in the state of Wyoming since I was 21. I moved here 4.5 years ago, and I just absolutely adore being in Wyoming, working on the fossils in this great state, and getting to chat with folks about how awesome our fossil history is.
And she has some up on the table.
And I have some up on the table here, so please afterwards come up and look at them. And one of the things that's so special about Wyoming is we have this incredible record of these very, very short global warming events called hyperthermals. And so this is depicting the Bighorn Basin across one of these hyperthermals. So here we are in the late Paleocene, like 57 million years ago, and then it gets really warm and it dries out, and you have all these animal invaders that come in and all these new plants. And then afterwards going back to like what it was in the Paleocene, but with new animals. So Wyoming is the best record in the world for this event, and I'm going to talk a lot more about it tonight. Okay, so of course, science is a team game. Lots of people who have been really influential in some of the work I'm going to show you all tonight. So a lot of this was done with Scott Wing, who is the paleobotanist at the Smithsonian. I've been working with Scott since I was in college. My lab group, some of you might recognize Esther, who was here about a year ago and told you about Eocene plant fossils in the Du Bois area. And then many, many, many students. And this right here, this is Sarah Fanning. She's an undergrad in my lab, and she's been helping a lot with the study of the second of these hyperthermal events. Okay, so here we are today, and I mean, that's the University of Wyoming, not here in Jackson Hole, but close. Okay, so this is what Earth looks like today. Maybe we're in a new geologic time period called the Anthropocene. And if we would go back in time, Earth would look very, very different than it does now. Okay, so first off, we could look at the continental configurations. We could see continents in different places as you go further back in time. And then also on these reconstructions, they show us about the extent of the ice sheets. So different climate regimes as well as we go back in time. So let's go back 1 million years to the height of the last ice age, and you would have either ice covering you if you're far enough north, or giant mammoths wandering around. That's out at the Mammoth Dig site in South Dakota, which is just one of the most amazing places I've been to. Great, great tourist stop. 56 million years ago, so this is what we're going to focus on tonight, this very warm interval, the warmest time since the dinosaurs died. And those are some of the plants you would find in Wyoming. Then further back in time, so 200 million years ago, that's near the beginning of the age of the dinosaurs. Going further back in time, so 370 million years ago, that's when our first ancestors came up onto the land. And my fellow Penn Stater back there, so near Penn State, there's a beautiful fossil site called Red Hill that preserves some of our earliest limbed ancestors. Further back in time, so 600 million years ago, there's our earliest ancestor with the backbone, which was still living in the ocean. First chordate. And then going back, sorry, that was 500 million years ago. 600 million years ago, no complex life, lots and lots of bacteria, which on the bright side do photosynthesis and pump oxygen into the atmosphere. And then we'd continue to go back another 5.4 billion years, or 5.4 billion years ago, about the age of the Earth. Okay, so as I already said in my introduction, Wyoming is world famous for its fossils. And historically, these are the sorts of people who went out and collected fossils. They tended to be quite white and quite male. So in addition to my work on science, I've also done a lot of science advocacy. So I co-founded a nonprofit called the Bearded Lady Project, challenging the face of science. And we have a documentary film. You can see there's my movie star film poster. We're working on a long film, which hopefully PBS will pick up and everybody will get to see it. And then there's also a photography exhibition that goes along with it. And so we use this humorous concept of, do I need to put a beard on to get taken seriously, to try to get people to expand their ideas of what a scientist looks like, who can be a scientist. And hopefully now when people imagine scientists and paleontologists, they might think of more people who look like this. And I will note, we're still heavily white dominated in our pictures, but unfortunately, it's really pretty hard to find a female woman of color who's a paleontologist. But we are hoping to change that. Okay, so we have our fossil collectors. We have the amazing fossil record of Wyoming. And so this is Dr. Laura Vietti, who's our collections manager at the UW Geological Museum, provided this slide for me. And this is just our geology museum in Laramie. is it just our museum? I think it is just our museum. And it's all of the places that we have fossils from and how many different fossils we have. So you can see fossils all over Wyoming, and the color of those dots, which represent fossil collections, is based on our geologic time scale here. And you'll note lots of orange colors, which are the Paleogene. So 66 million years ago, the dinosaurs die, and then we have the start of the age of mammals, also the start of the age of flowering plants. So the Paleogene, 66 to 23 million years ago. We are going to focus on the earlier part of the Paleogene, the Paleocene and Eocene. Okay, so when I'm wandering around Wyoming, this is what I'm looking for. These are primo rocks to find plant fossils in. And so what we're looking at here, this is a sequence of fluvial deposits, so river systems depositing sands, muds, or okay, so up here, like we have channels, so that would be kind of where you had actually the stream cutting through and putting in those coarser grained materials. And then also note down here, oops, sorry, not so good at this. Well, that darker layer down there, so that's a coal bed. And so those coal beds represent places far away from the stream where it's very swampy and you have lots of plant material just building up and getting buried. So the coals are not the greatest place to actually find fossils in, but if you look in the layers just above the coals and look in finer grained rocks, you can find beautiful, beautiful plant fossils. So, okay, now we're heading into a lake system, but if we go down to southeastern Wyoming, to the Green River Formation, the iconic palms telling us it was warm. Traveling around other parts of the state and things that I have actually found. Unfortunately, I did not find that. But so upper picture from Bighorn Basin near Worland, we have these tropical bean trees. From the center of the Wind River Basin, these beautiful climbing ferns, which today you would find in places like Florida, Mexico, Central America. Continuing our trip around the state, so this is maybe my favorite fossil ever. It's from one of the Paleocene, Eocene thermal maximum sites near Worland. Oh, there we go. And you'll note it has all of those beautiful bug bites in it. Then other parts of the state, so west of Du Bois, where today you would just find conifers, we had sycamore trees. Shoshone, which today is a hot desert wasteland, 52 million years ago, these beautiful horsetails. So a very warm, very wet environment.
How long ago?
These are all, so they're between 56 and 50 million years ago. Okay, so very, very warm time. So here's our paleogeographic reconstruction of what the world looked like during this hothouse Paleogene. Okay, and it's only 56 million years ago. So continental configurations, somewhat similar, although of course India still needs to make the trip up and collide with Asia. But you know what really comes across here is no ice caps and these big inland seas. So very, very warm time. That's right, no Florida. And the scientific data bears out how warm it is. So this figure right here, this is one of the iconic studies done in the paleoclimate community. It's reconstructing Earth's average temperature from when the dinosaurs died to the present day, or I should say to the pre-industrial day. And the way they get this record, they look at the shells of teeny tiny organisms called forams, which live on the ocean floor. And you can look at the chemical composition of these forams, and that is related to what the temperature of the water they grew in was. And because they're on the ocean bottom, that's kind of tempered from short-term changes, so it's giving you this long-term average record of Earth's temperature. And so we see, it's pretty warm when the dinosaurs died. You hit this peak warmth between about 53 and 51 million years ago, and then sort of a steady cooling, but with wiggles, to pre-industrial times. We might also note, so during this time period here, you see some very, very jarring spikes in the curve, and that's these, well, that one is the biggest of these hyperthermal events, the Paleocene-Eocene thermal maximum. What I wanted to get across with this figure, so this is telling us that all of Earth was quite warm. And so we have on our x-axis is latitude, so going from the equator to the South Pole to the North Pole. And this black curve here, that's the modern day temperatures, so a very steep gradient as you go from the equator to our icy poles. But if we focus in on that orange line, that's the time period we're interested in tonight, you see it's a much less steep temperature gradient. So globally, very warm, very, very different from today. Okay, so a lot of my research, as John said, so I'm a paleoecologist, I reconstruct environments in the past, and I reconstruct organisms and their interactions. So I'm really interested in plants and in the insects that eat them. And so my science is driven by trying to answer questions about how temperature and carbon dioxide levels affect plants, insects, and their interactions. And then more specifically, looking at these hyperthermal events. So how do very abrupt changes in carbon dioxide and temperature affect organisms? And I write in all my grant proposals that, this is important because we're doing it way, way faster than Earth ever did it. So this is our best geologic analog for the present day. And we'll see that soon. Well, in my presentation, we'll see that. Okay, so first to get some of the scientific grounding and the hypotheses about what would you expect first as you increase temperature, and then we'll talk about as you increase CO2 levels. Okay, so first thinking about plants. Okay, so probably intuitive to you, up to a certain point, as you increase temperatures, plants grow faster. Plants do a lot of enzymatic reactions like photosynthesis, and those go faster when it's warmer. However, plants, if it's warm but dry, that's not good for plants. So think about all the water escaping through the leaves. So you don't want to heat it up too much because water will just get pulled out through that plant. So the hypothesis that we might have for plants as you increase temperature, I would think their growth would kind of stay the same. So they could grow faster because it's warmer, but they also need to be careful about losing water, and those two would kind of balance each other out. Okay, as far as insects, so insects are cold-blooded organisms, and they do better when it's warm. So their metabolic rates increase. That means they can grow faster. It means they're not larvae for as long, and that's when they're most vulnerable to being eaten by other things. So probably the populations should be bigger when it's warmer. Also, if it warms up enough, that means insects can go through their life cycle faster, and you could get more generations of insects per year. And we are already observing that in nature. So my hypothesis is that as you increase temperature, you should see more insects. And then last, thinking about the interactions between plants and insects, and especially the insects eating the plants, so herbivory. And so looking at the modern world, so first off, we see that the tropics are way more diverse than the temperate zone. So we might expect more types of plants and insects in Wyoming when it's warm than when it's cooler. A variety of modern studies have also shown that herbivory is quite high in the tropics. So maybe this is driven by the high temperature of the tropics. And that's one thing we're trying to test with this work. And then we know from looking at the studies of the Ice Age, the last Ice Age, and how organisms advanced and retreated as the glaciers advanced and retreated. And so we know organisms could move quite quickly across the landscape. So we would expect that in the past, things could respond to these climate changes. Okay, so again, increasing temperature, expecting more diversity, more herbivory, things able to respond to climate. Yeah?
Ellen, when you look at plants, wouldn't there be a difference? Don't you have two variables, heat and humidity?
Exactly.
And water?
Yep.
Can you talk about that a little? Because some places it's going to be dry and the plants will disappear.
Yeah. Yes, that is exactly what I would expect. And actually, if we look at the modern world, the availability of water is more important than the temperature in driving plant distributions. And so, so if it's going to warm up, you also need to have a good amount of water to keep things stable. And I think a lot of the changes that we'll see in Wyoming might, or at least in the Bighorn Basin, might have more to do with changing the water cycle when temperature changes. But one of the things we're doing that's super cool, so it seems like up in the Bighorn Basin, as it gets warmer, you shift into a seasonal precipitation. But now we've got a big project in the Hanna Basin in southeastern Wyoming, and it's coal beds all throughout. So we think water availability was probably quite high through this whole time period. And so we're super psyched to see what's going to be the difference between those two, but we're not there yet. Okay, so temperature is very well correlated with carbon dioxide levels. We know carbon dioxide levels were high in the Paleogene, and they skyrocketed during this hyperthermal event. So what happens when you increase carbon dioxide levels? Okay, oh, all the words came up at once. That's all right. Okay, so for plants, you know, carbon dioxide, that's their food. They're fixing the carbon, turning it into the sugars and the starches and their bodies. So plant growth would probably increase with increasing CO2, but their nutrient concentration decreases. So we know from growing a number of plants in growth chambers at high CO2 that they become less nutritious as you pump up the CO2. They're building their bodies more out of starches, and there's less nitrogen-containing compounds in their body. So nitrogen, super important. We eat our protein, either meat or legume form. So as plants are growing in higher CO2, you would expect them to be less nutritious. And there have been some really interesting popular science articles about how nutrient concentration in plants are changing and what we might have to look forward to in the next 100 years. Okay, as far as insects, to the best of our knowledge, carbon dioxide doesn't affect insects. Of course, insects, especially those ones that eat plants, are going to be very highly affected by the nutrient concentration of their food. So if plants get less nutritious, then insects would need to eat more to compensate for that lower nutrition quality food. And then sort of concurrently with that, so if food quality is down, then insects wouldn't be able to grow as quickly. Their development rates would go down, and potentially insect population levels would go down as well. So it seems like temperature and CO2 might have opposing effects on insect populations. Okay, but our hypothesis going in is that, so as you increase temperature, increase CO2, insects would probably need to eat more. Okay, so we go out in the field and test these ideas, look at these time periods where you have big changes in temperature and CO2 levels. And this is the fun part of the job. So going out to various parts of Wyoming, most of this work was done in the Bighorn Basin. And you dig, these lovely quarries. So this level right here that Jeff is at, that's where the really nice leaves are coming out of. So we expose down to that layer and then very carefully extract block by block, split those blocks, look for pretty leaves up there for you to see afterwards. So we go through a lot of rock. The ones we want to keep, we wrap up in our industrial strength toilet paper and take back to the lab. I have very strong feelings about toilet paper. There are ones you use for fossils and ones you use for yourself, and very rarely would you want to switch. Okay, so what we do, so we dig one of these quarries, we very carefully excavate what we call the pay layer, where all those nice fossils are. I'm an ecologist, and so I'm really interested in trying to identify every single thing there. So trying to get an unbiased view of what's the distribution of plants on the landscape. So we identify as many leaves as we can, hopefully more than 450. And that number actually just comes from, that's the number that my worst site had. So it would be good to have more than that. The reasons why it would be good to have more than that are then you're capturing the rare species, not just the abundant ones. And then also, I'm really interested in looking at the things that have fed on the leaves. And I have found that on average, like 35% of my leaves have traces of insect feeding on them. So to get a good sample of bug-bitten leaves, it takes a lot of leaves. And we don't collect them all. The museums would kill us. So a lot of it is just censused out in the field, but we make sure to take vouchers of every distinct plant species and then all the bug-bitten leaves. Okay, so unlike animals, plants are really hard to assign scientific names to. And the reason for that is, okay, so it's not hard if you're dealing with flowers, fruits, seeds. The structure of those are very well conserved within a species, within a family. So if you've got those parts, you can put scientific names on things. Leaves, that is absolutely not true. And I grew up with a mulberry tree in my backyard. We had leaves that looked like that, and we had leaves that looked like that, and it was on the same tree. So it's really hard to tie the leaves to a particular scientific name. But we feel pretty confident that we can play the matching game and we can lay out all the leaf fossils and sort them into species. And I will say with those mulberry leaves, even though the bulk shape was very different, if you look at the details of them, and especially the vein structure, they are quite similar, whether like that or like that. So we feel reasonably confident we can divide the leaves up into what we call morphospecies. So it's a species, but we're not going to tell you what its scientific name is, and we're not going to tell you how the different species are related to each other. So that's the Bible in my lab, the Manual of Leaf Architecture. It has the different characters you can use to describe leaves. So we use these sets of terms, describe the leaves, and then we can compare species that we find at different times in different places. Okay, then turning away from the leaf material itself and looking at what's missing. So this is one of my favorite photos that I've taken. This is in Panama, looking up at the canopy and just beautiful, beautiful bug-bitten leaves. So insects have this wide array of ways that they eat or otherwise use plant tissues. So from some work that we've done in Panama, looking at insects eating the leaves, you can see ones that are chewing the leaves from the outside. And then we also have types of insects that access the leaf from the inside. So I grew up in the Midwest, lots of oaks, and you have these tremendous, they're called galls. So you might initially think it was a fruit, but then you're like, wait a minute, it's an oak. Oaks have acorns. What we're looking at here, so the female insect laid an egg inside of the leaf, and something about that laying process, either hormones given off then or hormones given off by the developing insect, caused the leaf to build this tumor. So we have a tumor on the leaf. The insect larvae is very happily inside of the tumor, protected from parasites, protected from predators, eating the inside of the gall, and then eventually hatching out as either late instars or adults. Here's another example of a type of gall. And then we have the leaf miners. So female insect laid an egg inside the leaf, and then that larvae stayed inside the leaf, eating and pooping its way through. You can see the poop trail gets bigger, and then eventually hatching out either as a late instar or as an adult. Okay, so if we were to wander around a deciduous forest, we would see these things today, and we see them preserved as fossils. So the question that I'm most commonly asked is, how do you know an insect did that? And I tell you, well, I know what happened while the leaf was alive. So we see these dark rims around the damaged area. Leaves can't regrow tissue. What they do instead is just put a thickened rim around the damaged area to keep more nutrients, water from leaking out there. And that thickened tissue is preserved a little bit differently in fossils. And so if we see that darkened rim, we can say, yes, the damage happened while the leaf was alive. If we don't see it, then we don't count it. And then it's just an assumption that an insect did that. Today, insects do most of the damage on leaves, but not all of it. So undoubtedly, slugs and other animals were doing some of this feeding as well. Okay, so we have things chewing the leaf from the outside, things that chew the nice soft leaf tissue and leave the veins behind. Here's some of those galls, some leaf mines. Okay, and on very rare occasions, we're lucky enough to catch the insects in the act. So what I'm gonna be talking about tomorrow, this amazing fossil site in Ethiopia, so about 22 million years old, and if you put it under the right type of microscope, shine the right type of light at it, you see these beautiful little scale insects preserved on the leaf. They would have been sucking out the good nutrients from the leaf. Now, on other occasions, we don't have the insect preserved, but it's a very stereotyped type of damage. So we can link a type of damage we see with an insect. So over here, obviously, this is not a fossil, green, squishy. This is a modern Hispine beetle feeding on a ginger leaf. And this is also looking at a modern specimen. And you see this very characteristic feeding pattern where the larvae scrapes along the surface of the leaf in these sort of elongate tracks, and it avoids the veins. Okay, gingers, super common in Wyoming in the Paleogene, and we find tons and tons of Eocene ginger leaves with that same damage pattern on them. And we can even go back before the dinosaurs died in the Cretaceous. Scientists have found these ginger leaves with this damage. So that's pretty amazing. For more than 66 million years, you've had this insect group eating this plant group. And think about all the coevolution occurring between these two groups. Okay, it's almost Halloween. This is my favorite science story ever. So not just plant-insect interactions, but scientists think that maybe they even see plant-insect fungal interactions. So we have our zombie ants with death grips. So this is known from, I believe it's modern Costa Rica. No, Thailand, sorry, modern Thailand. So there's this fungus, and I'm not even gonna try to pronounce it. And they're fungal spores spewing around in the atmosphere. Some of them land on this particular species of ant. And they get into the ant, they take over its brain, and they tell the ants that it wants to go from living in the canopy to going on the underside of a leaf 25 centimeters above the forest floor. That's optimal temperature and humidity conditions for fungal growth. So the ant goes down, grabs onto the bottom of the leaf, sucking some nutrients out of the leaf to keep it alive while the fungus eats the ant from the inside out. I know. And then what we're looking at in this picture right here, so this ant is long dead. The fungus, so a capsule grows out of it. Here are those fungal spores ready to explode and infect the next generation of ants. So there are all sorts of wonderful stories about organisms that take over the brain of other organisms. And this is my favorite because from Messel, which is an Eocene fossil site in Germany, people have found feeding patterns, feeding-esque patterns that look very similar to what those ants did once they'd been taken over by the fungi. So the hypothesis is that you had this tritrophic interaction in Germany. I think Messel's like 45 million years ago. So there's our fun Halloween story. Halloween is my favorite holiday. Okay, but so we have these cases where we can say what was doing the eating. A lot of the time we can't. So we just see, oh, here's beautiful leaves. This leaf, that button. This leaf right here, it's been chewed. That leaf right there, it's been skeletonized. So tissue eaten, but vein left behind. But we have no idea what insect did that. But there are things we can do. We can measure the amount of area that was eaten by insects. And this is much, much easier now that we have tablet screens that you can draw on and trace things. We can also count the number of different damage morphologies that we see. So the other Bible in my lab, the damage guide. So just laying out what are different morphologies of damage, and then you can see how the number of morphologies of damage change through time. Okay, so how do these damage types relate to insect diversity? They're pretty nicely correlated. This was some modern work that we did down in Panama where they have these wonderful things called canopy cranes. So 2 different canopy cranes in Panama. So there's, it's a general construction crane, but it's got like a box on the end of it that can take scientists up and it can deposit you in whatever tree you want to be in. So there we are in the box, zooming around the canopy. Well, not really zooming, but being slowly lowered into different trees in the canopy. Oop. Oh, wait, I'm pushing the wrong button. That's why nothing's happening. And of course, you have adorable animal encounters. Okay, so this was a grad student at Penn State, Monica Carvalho. This was her master's thesis. And so what she did, she took this very high-tech kite-like thing, and she would get lowered into different trees. She would beat the branch, hold the kite under it, and so then she could capture whatever insects fell out. Then she would pull leaves from the plant, stick them in a bag with each individual insect, and record what feeding traces each insect made. She identified all the insects, figured out how many species of insects she had, and then how many types of damage were made in the lab on in these bags. Okay, so here's her data. So each dot is a plant species, and it's our x-axis is how many insects she captured that ate that plant species. The y-axis is how many types of damage she saw. And then there were 2 different sites, so hence the blue dots and the yellow dots. And so it's not a perfect correlation, but it's a very nice positive relationship. And so we can be confident that as damage diversity is going up, the diversity of insects is also going up. So nice ground truthing. Okay, so with that somewhat lengthy introduction, now we're finally going to get to Wyoming and talk about what happens during these global warming events. Okay, so this was that curve I showed you of ocean bottom water temperatures. This over here are our temperature reconstructions from the Bighorn Basin. And we have the same pattern that you see. There's a kind of a gradual warming through time. Here's that Paleocene, Eocene thermal maximum, so that right there. And then right here, the EECO, early Eocene climatic optimum, that's that sustained warming 51 to 53 million years ago. Okay, oh yeah, so that hyperthermal event, that PETM, very, very obvious in an ocean core. You go from nice carbonate material to these brown clays. Also very obvious looking at the chemical composition. So on the left side, that's looking at the oxygen isotopes, and that's the same thing that's being done here to get temperature. So there's that big shift coming from this interval right here, and that tells us it's a warming event. Then our other plot here, this one, that is carbon isotopes. And you see a big change there. And what that tells us is there's this big perturbation to the carbon cycle. And I saw on the schedule, you guys had Gabe Bowen who came out a while ago, and maybe Gabe talked about this. He's one of the big geochemists who's been working on this. So the carbon isotopes profoundly change during this global warming event, and that tells us there was this big burp of light carbon into the atmosphere. Okay, where did it come from? Short answer is we don't know yet. Various hypotheses. One is that it came from the ocean floor where you have methane stored in solid form. Other ideas is caused by volcanism, by burning of shallowly buried peats and coals, maybe a combination of the above, and thresholds. So this was a really interesting study that was done on the New Jersey coastal plain. And so just what I want to get across here. So this curve right here, this is telling us about when the carbon isotopes change, and that's happening up here. This is a microorganism that likes warm conditions. So note that it shows up before the carbon isotopes change. And then this is a geochemical way to tell temperature. And note that that also changes before the carbon isotopes change. And so that suggests that there was warming before the carbon burp. So Earth hit a threshold, carbon burped. We're just gonna skip that. Okay, so what do we think happened? Approximately 4,500 petagrams of carbon were released. And this is somewhat comparable to what's left in our fossil fuel reservoirs, assuming we don't find big new amounts. Carbon dioxide levels increased 2 to 5 times. Oh, and I should say that carbon was released over something like 1,000 to 20,000 years. So CO2 at least doubled, ocean acidification, warming, 4 to 8 degrees Celsius, depending on where in the world you are. And then the entire PETM lasted about 200,000 years. Okay, so what did that do to the biota? Quite a lot. So those little microorganisms on the ocean floor had a big extinction. Interestingly, ones in the surface waters actually did pretty well. They had a big diversification event. This was a time of tremendous reshuffling of mammal distributions. So all of these things from Asia, primates, horses, and other odd-toed ungulates, even-toed ungulates, they all have their first appearance in North America during the PETM. In addition to lots of things showing up, body size shrinks. So you go from having dog-sized horses to like cat-sized horses. So getting back to that plants being less nutritious, one hypothesis for why you had the shrinking mammals might be that they didn't have as good a food source. Okay, let's talk about the plants and the insects, which is my work. So Paleocene, Wyoming is somewhat boring. We have not very many species. We have a lot of conifers. So this is dawn redwood, the metasequoia. This is the Katsura family. It's a nice ornamental plant, today only native to Asia. And then here's the sycamore family. Then we go into the PETM and diversity increases and we have all these legumes. So this is the most abundant thing in the PETM, it's a legume. This is the second most abundant thing, it's a legume, some other legumes. That's the sumac family, and that's just something that looks really tropical. Okay, and then after the PETM, it looks very much like the Paleocene again. So not very diverse. The conifers come back, the sycamores come back. So there's my picture version. There's the data. So looking at these 91 different leaf types. And you see, so not very many things go extinct because of the PETM. Here's these guys that just show up for the PETM. A very few things that don't seem to care and get all the way through. A few things come in afterwards. And then lots and lots of things that we see before and afterwards, but not during. So we think this is a migration story. It gets warm, things come up from the south, stay there as long as they can. The things that were there either went north or went up into the mountains, and then temperature goes back and everything goes back to where it used to be. Okay, as far as the damage, so during the PETM, lots and lots of different types of damage, and so some pretty pictures. So we have some leaf mines, these beautiful big holes chewed in the leaf, various types of galls. I'd said earlier, on average, like 35% of leaves are damaged at a typical site, 57% here. Okay, and so this figure, now this is putting, so I looked at these 9 different floras in the Bighorn Basin. The dotted line is the PETM. There's these points that are right before the PETM, and then that point during the PETM. So our temperature curve for reference, this is looking at how many plant species you would see. And then let's just focus in on the black squares here. So very high damage diversity during the PETM, but also up here during the early Eocene climatic optimum. Okay, so what's driving those trends, and why is there so much herbivory in the PETM? And you might be able to guess that, my introduction alluded to it, but let's consider all the factors. Okay, so one possibility, so we looked at how plants are changing, and there's a big turnover in plant species, and maybe those new plants are more cheaply constructed or easier for insects to eat. Okay, another idea is maybe it has to do with plant diversity. So more plants, more food sources, maybe more insect species, trying out those different food sources. Okay, then we have this idea of migrations, so diverse insect populations coming up from more southerly latitudes. And then thinking about that CO2 again. So plants being less nutritious and insects needing to eat more. Okay, so I can test that first hypothesis about things being being more cheaply constructed. And so if we think about a leaf, the thicker and tougher a leaf is, the bigger the petiole you need to hold that leaf on the branch. And scientists have gone and they've found a very nice correlation between what's called leaf mass per area, so how thick and tough the leaf is, and then the size of the petiole normalized for how big the leaf is. So I have a fossil leaf, I can measure its area, I can measure its petiole, and then I can back out this leaf mass per area. Okay, so I did that at my 9 sites. And I get a big mess. So yay, I can rule out that hypothesis. There's not any sort of trend in leaf toughness through time. Okay, then these plots, so I'm looking at plant diversity on my x-axis, and then either percent damage or damage diversity on my y-axis. And those R-squared values tell you how good the fit of a line you would put through the points. Those are terrible R-squared values. So, I mean, 0.38 is not bad, but we can do better. It doesn't seem like plant diversity is driving things. So I can rule out those hypotheses, and then I'm left with thinking about temperature changes and CO2 changes. Okay, so now look how much better my R-squared values do. And this very, very strong correlation on this plot right here between temperature and damage diversity. So as it gets warmer, you have more diverse insect populations. My plot over here on the left, looking at damage diversity versus the damage frequency, those R-squared values aren't quite as good. And so that got me thinking some more, like, you know, what's going on there? Oh yeah, so a lot of this work was my PhD. You have a finite amount of time to finish a PhD. You do not have armies of students who can help you finish your PhD. So as I was doing my PhD, in the back of my head was like, I feel like the PETM leaves have way more area damaged than any of the other sites I've looked at. I'm gonna file this away, and when I become a professor, then I'll have undergrads help me with this. And I did, and they're wonderful and enthusiastic. And this is the equivalent of washing beakers. You got to work your way up. You've got to measure those leaf area damaged. So thanks to a bunch of wonderful undergrads, we measured what % of leaf tissue was eaten at, we only did these 4 sites from the Bighorn Basin. And then these are other sites that other people had done and had published. And you can see almost twice as much area damaged in the PETM as at any other time. So my gut instinct was right, but then I was left with, well, okay, why? And why especially? Because this site right here, that's my PETM site, it was just as warm as this site right here, which is the eco. And if it was just as warm, then CO2 levels were probably just as high. Okay, so this is data looking at the bulk floras. I was lucky. There are some plant species that are in the PETM and in the eco. So this thing, Populus, cinnamomoides, aspen family. So we had that species that we could trace through time. And so I think that, sample size of one species, but I think very interesting comparing this % of leaf area damaged. And again, we see that PETM being almost twice as high, or in this case, more than twice as high, as during this early Eocene climatic optimum. So, this took a lot of puzzling and thought, and I don't know if I have the right answer, but this is the best I can do. Legumes. So in the PETM, we have all those legumes, and you don't see those in the early Eocene climatic optimum. So if anyone here is a vegetarian, you probably eat a lot of legumes to get your nitrogen. So legumes have a symbiotic relationship with nitrogen-fixing bacteria. They have higher nitrogen levels. So my idea, and this just documenting, so these are these other Bighorn Basin sites, and you see lots and lots of legume leaves compared with other things. So my idea is that you had, these very nutrient-rich plants that were buffering the decreased nutritional value of the high CO2. So these plants got more available, or more nitrogen to be available in the environments, which other plants could then take up, which could sustain bigger insect populations. Okay, so I made these beautiful graphs, and I'm very proud of on this one. If you put 2 data sets on a graph, then it looks like you have more points. But I admit that to you all. So one of them is looking at counts of leaves, and the other one's looking at areas of leaves. And you see a correlation based on those 5 points. Definitely more work needs to be done looking at other sites, seeing how important legumes are and if that's impacting the amount of damage and the types of damage you see. So pulling this all home in a flow chart. So during the PETM, you had these changes in temperature, changes in carbon dioxide levels. So temperature generally having a strong positive effect on insects, letting them grow faster, eat more, so you would expect more total consumption. But then CO2 having that opposite effect, so plant nutritional quality goes down. These things one might all expect to go down, although maybe that goes up. But with our friends, the legumes, that helps to buffer this, giving bigger insect populations and more consumption. So that's my idea. More work to do. Okay, so I promise you I'm really close to wrapping up. So that was the PETM. We now know that wasn't the only one of these hyperthermals. There were a series afterwards, and you can kind of see one right there, but it's a little wiggly and the dots are too close together. But if we were to pull that curve apart, look in the ocean or look in the Bighorn Basin, and you would see these spikes in the carbon isotopes. So we have now discovered the first plant site from the second of these hyperthermal events, the Eocene thermal maximum 2. And this is what Sarah has been working with me on. So, you know, it's kind of fun doing science in the age of smartphones. You can send your colleagues coordinates and they'll tell you where on the carbon isotope curve you are, and then hopefully you can be confident that you are in that hyperthermal. And actually, I think that some of these, that top leaf is up there, so some of these leaves came with me today. So we seem to have this very interesting mix of normal Paleocene, Eocene things, but we do have a lot of legumes at this site. So this event was not as extreme as the PETM. CO2 levels didn't change as much, temperature didn't change as much. And so you seem to get this mix of regular Eocene things and then maybe hyperthermal things. And so this is preliminary data. So what you're looking at here, each dot is a plant site. And they are plotted using this ordination technique. And basically, the idea here, so you have the sites, they have all of these different species at them. And points that plot very close together have very similar species compositions, versus plants or sites that are quite far apart have very different plant species at them. And so our red points are the PETM sites. Blue and green are normal Bighorn Basin, Paleocene, or Eocene. And there's that ETM2 site that very nicely plots between Paleocene, Eocene, and PETM. So magnitude of change seems to be very important. Sarah's working on herbivory data right now, so we don't have that yet. Okay, so to conclude, so plant fossils are amazing, way better than dinosaurs. All this environmental information you can get out of them. I didn't talk about where our temperature reconstructions came from. They came from the plants themselves. So lots and lots of cool things to do with plants. Looking at insect herbivory, we get this, one of the only direct pieces of evidence of a fossil food web. And then we had this hyperthermal event, very rapid changes in temperature and carbon dioxide levels. So during the PETM, we had these big range shifts. And we're observing range shifts today in response to warming. One thing I really want to hammer home is that during the PETM, those rates of environmental forcing are 1/10 to 100th of what they are today. So plants and insects did just fine 56 million years ago. That doesn't mean the same will be true today. And then now looking at this ETM2 event, we see very clearly that the magnitude of the event is important for how much change there is. So the less severe events, less biotic change. Okay, so here we are today, our wonderful Earth. There is no planet B. But maybe we're interested in where we're headed in the year 2100. We don't have time machines, but we have shovels, which Kirk Johnson likes to call time machines. So we can go back in time 56 million years ago and hopefully prepare ourselves for where we might be headed, or at least mitigate where we might be headed. Okay, we'll just leave that up. So thank you all for your attention, and John said you guys are a great audience for questions, so I'm here till 7:30. Yes?
You had have that leaf architecture book, and I made a note of it.
Yeah.
Because I might like to get it. But
It's on Amazon. It's like 20 bucks.
I have been fascinated for years by the different shapes of leaves.
Yeah.
And I've never found anybody who could tell me why and what the different shapes indicate. Why do different plants use different kind of leaves, and what advantages do one leaf?
Yeah.
Can you say a little about
I
that?
can say a little bit about it. A lot of it we don't know, but so some of it is genetically controlled. And so, for example, if I look within the bean family, I'm only going to find leaves with smooth edges. Whereas if I look like in the birch family, they all have serrate edges. So now some of it is genetically controlled, but then also some of it is environmentally controlled. And so we think about leaves, those are the solar panels. So the plant's goal or the leaf's goal in life is to capture as much sunlight as it can, but then also to mitigate water loss. So thinking about like a tropical rainforest versus, say, the African savanna. So in a tropical rainforest, we have really big leaves versus you go to the African savanna and you have acacia plants, those teeny, teeny, tiny leaves. So in wet climates, you can have bigger leaves because water loss isn't a problem. Versus in drier environments, you have smaller leaves, so not as much water loss occurs. Thinking about also whether you have a leaf, an unlobed leaf versus a lobed leaf, that might also have something to do with water loss. So in wetter environments, you tend to have more unlobed things. Having those lobes, you get like tissue pointed out in different directions, but it's not completely filled in, so less water loss in those areas. I'm going to keep going, but then did you want to contribute to the conversation, or?
I just wanted to ask a question. How
Yeah.
do you pick the sites where you dig?
Oh, okay, let me get to that, but I'm going to do a little bit more here first. So also thinking about the way the plant constructs a leaf, it's a balance between, the leaf has to be held up. So if you have a floppy leaf, it's not going to intercept sunlight, and that's a poor thing. So plants have to devote energy to having like vein systems that can hold the leaf up and then also transport things to different parts of the leaf. So plants, so some plants, like especially in cooler climates where you have a short growing season, if you've only got the leaf for 3 months, you just put it out quickly. Don't worry about defending it well. So you tend to have like more loby things or smaller things, not these big things that need to be held up and have to be very well constructed. So I could talk for hours about this. As John said, so I'm 75% in a botany department, and so instead of teaching the wonderful general geology class, I have to teach gen bio, and we go over and over again in class, structure determines function. So the structure of the leaf determines the function of the leaf. Okay, back to your question about how do I pick my sites. So it's a combination of what's available, but then also what science am I interested in. So, like all paleontologists, if someone calls me up and says, ooh, I have wonderful leaves on my land, and then I will go and check it out, and if they're good, you're not gonna waste good fossils. Hopefully you can figure out how old they are. Great student projects out of those. But then there's also okay, here's the science that I'm interested in. And so I really wanted to work on warm times in Earth's history. And so I look at like geologic maps and see, okay, where are early Eocene rocks deposited? And I'm interested in either lake deposits or river deposits. And then for a lot of the work that I do, it's really important to know how old things are. So I like going to places where geologists have worked. Geologists are vertebrate paleontologists. So geologists who have looked for ash layers given precise radiometric dates of different rocks. The vertebrate paleontologists are in fact useful because a lot of our biostratigraphy comes from mammal fossils. So you have these mammals that are only around for a short period of time, and which one you have tells you what part of time you're in. So doing this study, looking through the Paleocene, Eocene, trying to track down where the PETM was. And so I will say, so the PETM was first documented in the Bighorn Basin in the early 90s. And Scott Wing, my collaborator at the Smithsonian, since the early 90s, he has been looking for plant sites in the PETM, and it wasn't until 2005 that he found the first good one. So, Scott spent hours and hours and days traipsing through the Badlands in the areas that he thought were PETM, and it took quite a while to finally find it. Yes?
Do you use fossilized diatoms at all for any of the water finding you might do?
I do not personally. No. I know people do, but that's a whole different skill set that I don't have. Yes?
Ellen, as you probably know, high-yielding varieties of rice with global warming are becoming less nutritious.
Right.
I'm not sure exactly in what way, but I'm wondering, first of all, would you like to talk a little bit about that? And secondly, you had that very cool graph. It said bonanza on the upper point.
Yeah.
Do you remember that one?
Yeah, that's not too far ago.
It's about 6 back or 8 back.
That one.
Yeah. Now, that's got a really sharp slope.
Yeah.
Okay? So your R square is driven by those two extreme points. And does that slope give us hope in terms of positive steps one could take to improve nutritional value of things people rely on today to eat?
Yeah.
May not be a good question.
Yeah. Well, I mean, I'll first of all say, yeah, you're absolutely.
Repeat the question.
Oh. Oh boy. Okay. What's the question model?
Well, a couple of data people talking.
So, or here.
You try.
Oh. Okay. So, so people doing work today looking at nutritional quality of food, and it's been observed that a number of important human resources, including some rices, are becoming less nutritious. And then
The question is, what can one do?
what can we do about that? Yeah. Well, so this plot here, this has to do with how much feeding is occurring. And so I would say where we go from this plot is
The legumes.
yeah. The legumes are being preferentially targeted by insects when there aren't a lot of them. But then when there are a lot of legumes on the landscape, they're not being preferentially targeted.
So if one could increase the quantity of legumes, would that help?
Oh, yeah. I think so. That's what my data seem to say. And it would be great to do this in experimental plots and see if that raises the nitrogen levels for everybody.
Exactly.
Yeah.
If the high-yielding varieties of rice fail,
Right.
you're talking
Yeah.
immense food insecurity.
Yep.
famine.
Yep.
That is an interesting point. And you could make it in your graph, in
Yeah.
your proposals. You
Yeah.
could use that graph.
That's a really great idea.
Follow-up question. Why is the nitrogen so vital to nutrient value?
So many biologically important compounds, especially amino acids, require nitrogen.
Completes the protein.
Yeah. Yes.
Could you talk a little bit about how leaves are actually fossilized? Because obviously most leaves aren't. And well, with wood,
Yeah.
that's fossilized is often silicified.
Right.
But the fossils you have, are they carbon films? Are they, how just...
Yeah, absolutely. So the question was, how do leaves fossilize? So the main environments that preserve leaves are either lakes or river environments. Although we do have the very lucky case sometimes where if you had a big volcanic ash that could bury leaves, there's a place in the Bighorn Basin, it's called Big Cedar Ridge. It's about 71 million years old, so it's Cretaceous. This is a place that's got wing worked. And so you had this ash rain down and just bury the vegetation in place. And so it's so cool. Like you see the plants and their leaves. And so what Scott has done there is, lay out, what are they called? It's like a picture frame, but with string across it, and so marking out areas, and you lay this down, and then you can count how much area is of the different major plant groups. So in these like amazing scenarios, you get like the forest preserved, or well, not the trees, but the understory preserved by the ash raining down. The other stuff, so say you've got this lake and the trees are growing around the lake and the leaves fall off. If they sink to the bottom of the lake, if it's not a lot of oxygen at the bottom of the lake, so nothing can live down there, nothing can eat the leaf material, so the leaves fall into the lake, get buried, and we have our fossils. A lot of what I've found is looking at floodplain deposits. So you have the river, and then away from the river, you have your plants growing. And then periodically, the river floods. And when the river floods, it's not just water, it's mud. And then those flooding events bury the leaves that are on the ground. As far as what's still there, so that depends on a number of things. It depends on what, how do I say this? Like how much degradation has occurred, whether the leaves are heated and pressured, if like you've got various water moving through it, breaking things down. So in some cases, you don't see any leaf material. You just see like imagine, you have the new concrete poured on the sidewalk and the leaf falls and then someone pulls the leaf off or the leaf decays away. But then there on the concrete, you see the impression of the leaf. So we call those impression fossils. The ones that we have up here, all right, well, I'll just hold one up. Okay, here, right, maybe this one's the easiest to see. So you see a nice dark leaf on a light rock. So this is not an impression, we call this a compression fossil. And so some of that leaf material is still here. Some of that carbon is there. In some incredible circumstances, like the Lagerstätte where we work in Ethiopia, you actually have the waxy coating on the leaves preserved. So the leaf cuticle. And if you have the leaf cuticle preserved, you have the leaf epidermal cell structure. So if you come to my talk tomorrow, you'll see amazing pictures of 20-million-year-old plant cells. Well, it's not the cell that's being preserved, but it's the imprint of the cell on the wax.
Wow.
So we have time for one more question.
Okay.
Do you find insect fossils?
Very rarely. So the rationale for starting looking at herbivory, that was my PhD advisor, Peter Wilf at Penn State, and then Conrad LaBandera, who's a fossil insect scientist at the Smithsonian. And so insect fossils are awesome or non-existent. So you have places like the Green River Formation, where you have those beautiful, beautiful insects. You have places like amber deposits where, oh my God, like I was at Kansas working with a colleague, and he's got 120-million-year-old pieces of amber with these beautiful insects in them, and you can see every hair on the insect. Amazing. So you have these exceptional deposits that have insects, but they're few and far between. And so in order to understand how insects are changing through time, Peter and Conrad decided to look at the herbivory on the leaves. Leaves are way more abundant.
So before we do the chair. And I want to add that 12:15, show up a little earlier at the Senior Center tomorrow, and you'll learn not just about fossils and plants in Ethiopia, but you'll actually get a little Ethiopian
Yes.
culture.
And they have a salad bar as well as a hot meal, so.
Right, very good food.